Hola y bienvenidos al seminario de divulgación interdisciplinario, más que nada dirigido a estudiantes de matemáticas aplicadas para que puedan tener una idea acerca del futuro académico y laboral y todo. Okay, so today our speaker only speaks English, so I will switch now and I will give a brief introduction. So he is Haralampos Hatzikirou, also known as Babis. And so he did his bachelor in mathematics in Thessaloniki, if I'm not mistaken. And then his master's, very good. Then his master's in Germany at Technische Universität Kaiserslautern, I think. And then his PhD in Technische Universität Dresden. And then a PhD in Texas, right? University of Texas in pathology. Yeah. Nice. And then he started as a group leader at TU Dresden, and he's also a professor at Khalifa University at the Arab Emirates. And he will give us a nice talk on cell decision making. So please, Babis, start.
So thanks a lot, Jose, for the nice introduction. I mean, with Jose, we know each other from my Dresden times, and we collaborate since 80s, so it's very nice. I mean, I wish this would have been, from close enough, but what can we do? Now we have to do it online. So let me share my screen. Do you see my screen? I guess yes, eh? Can you see me? Can you see that? Jose, I cannot listen to you. Yeah. Okay,
we can.
Yeah. Okay, good. And now the full screen. Okay, perfect. So again, thanks a lot for the nice introduction. And today, what I'm going to talk to you is a theory that me and my group are trying to developing about cell decision making for multicellular systems. However, I will start a bit historically, which means that I will give you how I ended up going towards this direction. And my first problem that I was dealing, and I still deal with, because we never solved it, it's cancer. So, yeah. So, and cancer, actually, of course, everyone knows a bit about cancer, and, the perception when someone thinks that has a cancer is to go to the doctor, right? And by going to the doctor, then the doctor will run some tests, like an MRI scan or, a biopsy. And the following, he will make a diagnosis, propose treatment. Then after the execution of the treatment, a follow-up exam will happen. And of course, the doctor needs to predict, again, the survival or, recalibrate the therapy. which in principle, this is not an easy thing. Although doctors and like clinicians, they do it all the time. However, I will show you why it's not an easy thing. And this is because there are these three challenges. So first of all, as you might understood, I mean, a lot of times you speak about medicine like big data, we have enough data. The truth of the matter is this data, especially for patients, like especially in cancer patients, they are not very frequent. So you don't have real-time data. You have sparse data in time. So for instance, you cannot make a magnetic resonance, every day. It's too expensive even for the insurance. Secondly, these data are noisy snapshots in space and time. So as you might observe, let me hear my, no, I think laser pointer, yeah. So if you think that I have here my biopsy, I mean, it shows a constellation of cells and what these cells are doing, but this time point. And, this is like, we don't know if this is tells you anything about time. And we'll see it in a while. And last but not least, we don't know all the biological mechanisms. We know partially something, but not everything. So the point of the mathematician that comes here, like me, is how can we connect the dots? And in order to motivate you, as I told you before, I mean, we have this, let's assume you have these biopsies and, you have the cells that they are doing something at that point. However, let's motivate it in a different way. Let's assume the cells are football players. And what I'm going to do now to you, it's like I'm going to show you some photos of football games, and you might try, I don't know how interactive can it be here, but okay, if it's not too interactive, I will play the role of interaction. So the thing is that I will show you some photos, and then you might try to make a diagnosis, or you might try to think about what is happening in the future, so like a prognosis. So let's assume we see this photo. What do you think is happening here? So you see players celebrating. So in principle, the most obvious assumption is that this will be, they scored a goal. And in principle, this is true. Okay, now I think I have to return this to my pointer. Let's remove this. So as you see here, this is like the players score a goal, and of course the players, they are very happy about it, right? I mean, that's the proof. And then you will see very soon enough this instance. Yeah. Okay, so now the second thing I want to show you after you get convinced that this is the game, I mean, yeah, this is the game, right? So after I show you this is the game, then this is like, we were correct with our diagnosis. So we said, okay, what we assumed, it was correct. However, let's see this photo now. And in this photo, you see a player cry. So usually, when I'm asking what's happening here, most likely people think that it's something negative, right? I mean, they lost a goal or they lost the championship or whatever. The truth of the matter is that in this case, they just scored a goal, right? And because the player was very much intense, and then he cried. So the point is now this image told us a lie. So what we saw in the image didn't tell us the correct story. So the diagnosis was false, right? Okay, so this is, let's say, with the diagnosis. Yeah, I mean, you got convinced that this is a goal. Let's try now to make a prognosis. So to see to predict what's happening the next time. So we see this image. So what do you think is happening here? So you see the goal is the ball. It's, by the way, can you see my cursor when I move it so I don't switch it all the time to the laser?
Yes, we can see it.
Okay, excellent. So the goal is the ball is crossing the line. So most likely this is a goal, and what is going to happen is that in the next few seconds we're going to see a kickoff, right? However, this actually comes from the very well-known game between Germany and England in 2010, where this is this ghost goal, and Lampard, he was shooting, and then somehow it goes the ball in, but it never counted. So in the next times that, let's say, our prediction again, the image told us not the truth, correct? So our prognosis was false. So to cut a long story short, I mean, with these toy examples, what I wanted to motivate you is that if we see an image, if we see static data and snapshots in time and space, they might not tell us the full story. So we need to understand more about the dynamics. And this is actually, in order to understand this, we need to understand how cells make decisions, because as the players here, they were making a decision to do something, the cells, they do it as well. And let's go back into our sketchy cancer here. Let's assume now we have a cancer cell, this blue cell, and it communicates with the surrounding where there are different cell types. And, they send signals to this cell, and then, of course, it changes its phenotype. Phenotype, it's a buzzword for behavior in biology. So it changes its behavior, let's say, and becomes, let's say, from a blue pink. And then, but this doesn't come alone only by the change of the color, but maybe, this cell now changes also what it transmits to the other cells, to its microenvironment. So then it induces at the same time changes the microenvironment. And there's this dynamic dialogue on and on. And this, so between the cells, the cancer cells in that case, and the microenvironment. And this is a definition of a complex system. And what do you know about complex systems? What math tell us about complex systems? They tell us that we can have emergent behaviors, or we have the emergent organization structures. And actually, for instance, here, as I said, this is a cancer cell, the other cells can be epithelial cells or immune cells. This is how we like to understand the tumor, as an emergent behavior of a complex system that it's not doing, of course, what it's doing properly. And you might think this is very new, but it's not really new. So already in 1962, there is this Dr. Smithers that he was saying that cancer is no more a disease of cells than a traffic jam is a disease of cars. A lifetime study of internal combustion engine would not help us to understand our traffic problems. So he already recognized that looking only into the cell properties is not enough. We need to see how the interactions are taking place, right? So, as I said, we're going to talk about these decisions of the cells. And in principle, there are two types of decisions of cells. And I want to put the slide here. Of course, not everything here is relevant to the cancer, but still, this is important to understand that we have two types of cell decision making. One, it's called cell-fate determination, and it's in principle a reversible process where the cell starts from stem cells, and then, it undergoes different differentiation steps to committed stem cells or progenitor cells, and then it gets differentiated. What it means differentiation in biology? It means specialization. So stem cells, they are pluripotent. Actually, they are non-specialized cells that can become anything in principle if they find themselves in the right microenvironment. And then they will be specialized maybe to skin cells, to heart cells, gut cells, whatever, name it. So that's one process. But of course, we have reversible cell decision-making cells, which is called phenotypic plasticity. And again, phenotype, as I said, is behavior. Plasticity is the word for being very easy to change. Let's say, shows how easy something to change. And in principle, this is something. It's something that follows a microenvironmental cues. So the cell changes the behavior, but when the stimulus is get removed, then it goes back, right? So these are the two, very simple, these are the two cell decision-making cases that we're going to talk about here. But for the cancer, the most relevant is the latter. So here I will focus with the first problem that historically I was dealing with, which is what we call cancer cell migration proliferation plasticity. So in principle, this plasticity, it's a phenotypic plasticity between two phenotypes, migratory and proliferative, and simply has been known as a go or grow. So this actually tells us that the cell can become motile, and then if the microenvironment changes, it can go to proliferating state. And of course, this can go back. So you can see that always this type of mechanism, you can see them as a kind of energetic payoff, right? I mean, that, the cell wants to invest the energy, and all these processes are pretty expensive energetically, and then the cell would like to invest their energy optimally. So interestingly, although I started this thing with 2010, the concept was existing already in 1996, although it was not really in the mainstream. And then this has been discovered firstly in gliomas, at least observed. And however, and this is actually where we took it up. But however, after a while, it has been found out that breast cancers actually, they might exhibit this type of plasticity. Or melanoma, also, they have this switching between proliferative and invasive states. And most interestingly is even, this is in 2016, this review, that this is also a process that even healthy developmental cells, they might follow, or actually if they want, so if they go to the quiescent state, so this is actually the cell cycle, and the cell cycle actually is the process of cell being divided, but when the cell cycle is arrested, which means stops, right? Then the cells, they can go invasive, but when they are stopping invasion here, they can proliferate. So there is a kind of general strategy in cells, and we're going to try to analyze this phenomenon in order to be able to understand the problem of glioblastoma, of GBM. And in principle, what we want to understand in this problem is the problem of recurrence. So what does it mean? So glioblastoma tumors are brain tumors, as you might observe here. And actually, they are the most aggressive type of glioma tumors. And this type of tumors actually, as you see, even actually, even if you remove them surgically and you follow it up with radiotherapy or chemotherapy, it's almost sure that this will come back. So for instance, this is a very interesting sequence of MRIs. So you see, this is the patient that he got removed cancer. This is this hole here. But then in 3 months, the cancer has been coming back almost in full. And in 6 months, it's almost double. So really, this is a pattern that you still observe, and very few patients can escape this pattern. So the question here is now, so the first question, one of the first questions we had was, how can we identify the mechanisms that they are, I mean, if this is actually true, if we can see it in experiments? That was the first question, right? So then we took these in vitro experiments, these are the experimental pictures where they were letting multicellular spheroids, glioma spheroids, to grow. So this is a 2D cut of 3D colonies. And you see what the experimentals were doing, they were segregating here the dense core with the front. And then what they were observing that in time, this invasive rim, and they saw this divergent behavior. To cut a long story short, we wanted to know what are the mechanisms, so we made a model. So this is a lattice gas cellular automaton model. It's a cellular automaton model. It had 2 states, proliferative and moving states. But in the moving states, you couldn't proliferate. And when the cells were resting, they could proliferate with a certain probability. And then we assumed a Markov process that it was controlling the switch of the behavior between the 2 phenotypes. And in principle, what we found out is that the only way to achieve this divergent behavior was to assume proliferation. I mean, we scanned the whole parametric space, and the only way to achieve this behavior was if we would have assumed that the resting states are proliferative and the moving states are not. And also the other important thing is, okay, we found also that the cells could be repulsive in order to produce another this radial persistency that they observed during these experiments. But also another important thing that I want you to keep in mind, and keep what I'm saying now here, is that this phenotypic switch is descent dependent. We could feed a function, but let's see if this function is, let's say, validated in other experiments. So we went to another type of experiments. This was like 2D growth in vitro plating of glioma cells, where they were seeding the cells in the middle and they were letting them migrate. And because this was a short time, you see, it's like 2 days experiment. you saw they could observe, these radial density profiles, and then you see the classical exponential decay of radial profiles. But then, you have something that's a bit non-trivial. And then, since we had here no proliferation, because the time of the experiment was too short, um, we had not a go-grow model, but we had a go-rest model. And then we were able to fit the experiment. I'm not going to details how we fit the experiment, but the interesting thing is we found again that this phenotypic switch was tumor density dependent. However, the function was not exactly the same. Also, another interesting thing is that we found out that this kind of behavior was, this type of separation of two phenotypes was producing this anomalous diffusion. But this is another story here. But they keep this in mind now that still in two experiments we found out that there is microenvironmental dependence in this switching. We could identify that tumor cell density is a good proxy, but the function we couldn't pinpoint that. So as mathematicians, typically when we don't know something, we can, make hypotheses. So we took our model again in 2015, and we said, okay, let's try to make a theoretical study in the sense that we take our go-grow model, and then we allow this time the cells to die. But on the other hand, we assume that these switching terms, these phenotypic switching terms, as you see here, they are complementary. So this is from motile to resting, and from resting to motile again. And we assume two types of phenotypic switches. The first type is when the cells are kind of social. This is what we call attractive. And then, increasing the cell's density, then the cells, they are becoming more probable to stop and proliferate. So they like to see their peers, and then they stay there and proliferate. They feel comfortable. And when they have few cells around them, they go away. Then we assume the complementary mechanism where we had the repulsive cells, the repulsive strategy of cells, where when they had a lot of density after a certain threshold, they were more like to go away, so they were more invasive. And when they were, like in a more dense free area, then they would like to stay there and proliferate. Interestingly, the monotonicity of the previous experiments was this one, right? So about the aggressive GBM. Keep this in mind. So now what we have done, we made an analysis of the model. So the first thing we have done, we made a formal mean field analysis where actually we just assumed that, there are no correlations, and the high orders can become powers of the first moment. And also we assume that the switching dynamics, they are very fast. And then we could map the whole problem into a mean field equation, where this mean field equation, it is reaction diffusion PD, a nonlinear reaction diffusion PD, where the reaction term now, it's a pretty nonlinear term, right? Because you have here the classical logistic growth, but this RS, it's following actually this hyperbolic tangent. And then the diffusion term also is estimated around zero density, but again, it's density dependent. So the interesting thing is that this system, we can find the exhibits bistability between rho and rho zero, which is the extinct state, and the rho star, which is a state of fully growth. And then this happens only for the attractive case, right? And for another condition of the parameters. And this actually, bistability, is what is known in ecology as the Allais effect. Allais is actually an American ecologist that he lived in the beginning, 40s, I think he was working, his works are mostly known, and he was postulating that there is a critical threshold in a habitat of animals, he was speaking about animals, that if we cross this threshold, the colony, the community will grow, and if you go below this threshold, this will extinct. This is what's happening in our case, where if we cross the threshold, the Allais threshold, the tumor will go to a fully blown growth where the proliferative cells dominate, but when we are below this threshold, because the migratory cells are dominating, then, and they don't proliferate, of course, then they might get extinct because there is a finite probability to die. And if we want to see that, and of course, as we said before, the other repulsive case is monostable. So it's only growth. So again, what we observed in the fully malignant GBMs, glioblastoma cells, they were exhibiting only growth. So to show something here is that, to show you something is like the initial conditions of the problem matter. This is, of course, the bistability, right? And then if we start with big tumors, most likely you're going to end up with a low probability of extinction. And we start with small tumors, we're going to start with have high probability of extinction. And there is this middle ground. This middle ground, the blue line, the probability of survival is kind of, bimodal. So you have like equal probable. And if we set now, interestingly, our initial conditions on this point, and we set up the simulation, exactly two simulations, exactly the same parameters, exactly same initial conditions, you're going to see the following situation. In some cases, you are going to see that these virtual tumors are growing. And of course, in some cases, we're going to see the tumors, these virtual tumors getting extinct. And this is just, the stochasticity where drives the system either to the one attractor, or the extinction attractor, or the growth attractor, right? It's the noise-induced phenomenon. So the question would be here, okay, this is nice theoretically, but where is the reality here? And interestingly, there were two studies following up. This was 2015, our study. 2017, it came up this study that they found out that the glioma cells, they are exhibiting the weak allele effect. It's actually, they don't go extinct like the strong allele effect we saw before, but they're growing very slow when they are low in numbers. That's an interesting story. And the other interesting thing is in 2023, there was this Science Advances paper where actually they found out there is an allele effect in metastatic breast cancers. And also it's based on the go or grow, right? So this is actually what you see here, the per capita growth in the speed. So you see there is an inverse relationship. And somehow, if you read the paper, you identify that there's exact statement. So it's very happy, that of course this was not glioblastoma, but still, we could see this allele effect depending on the go or grow. So now the question is again, let's go back to the GBM. And I had, when I was studying as my PhD, I had one question. Always I could see experiments that they were high grades, but low grades. I never saw low grade experiments. And I was asking the experimentals, why you don't do it? And they told me they are not reproducible. So this is true, actually. Then I could find also some, these are two indicative references in the literature that will tell us that, in the contrary to the high grade, low grade tumors are not easy to be established in an experimental setting. So a potential explanation is that this allele effect might be there. And then if the experimentalists, they are not really careful with these initial conditions, you might have sometimes something growing, some nothing's not growing. And interestingly, here we can conjecture from our theory that this kappa parameter, which is actually the slope of this tangent, hyperbolic tangent, can tell us, if we have this social or antisocial behavior of the cells, and this is one associated with the allele effect, and the other is associated with monostable growing tumors. So this also could define us like cell density as predictive biomarker. Of course, this is some work on progress, but this is something that we can propose with our theory. So what the model before was overly simple. The question is, okay, what can we do if we have more complex models? And I'm not going to stay into that now. We built up more complex models, and then we saw that this grow, of course, that is, we put in this case. So here we had also vessels, and that we had vaso-occlusion phenomena due to high density and pressure. So all these, they had effects in therapies. But what I want to tell you is that what there was always a problem until here. Even until that time, even until now, we don't know the exact function, right? So still we have some uncertainty in the mechanism, how this mechanism is being executed. And of course, there are data, and there are some molecular data. The point is not easy to model them, right? So not easy to write equations about RNA sequencing data. So at this point, now it's the second part of my talk. There was the following situation where I was asking, how can we circumvent this mechanistic uncertainty regarding the cell decision making? And this is where I formulated a different, this LEUP, where I'm calling, but we'll see what is that in a while. So this is just a small reminder. We have, as we said, the two cell decision-making processes, right? And the one is the self-fate determination, which is reversible, and we have the successive specialization of cells during development, and the other is definitely plasticity. Now, what we needed here is we needed to find, some formulation that we don't need to write explicit functions about how the microenvironment is acting on the cell. So this is actually the tricky point. And in order to do that, I came across these two things. So there are these on the left, I'm showing you a study, which is 2013, where these people, they studied cell differentiation. So that's why I show you the cell differentiation, because, the most of the data that inspired me about this theory was on cell differentiation. And then they said, like, look here, let's assume we have pluripotent stem cells, right? And then here we have specialized differentiated cells. What they measured is actually the entropy rate of the internal state of these cells. Actually, they were measuring, in principle here, the entropy of the underlying gene networks. And what they found out is that this entropy, actually, it's much higher on stem cells, and of course, differentiated cells, they have lower entropy states. And this makes sense, right? Because you can assume that stem cells, because they are multitaskers or they are pluripotent, they have access to all the parametric space. So the distribution is rather flat but on this phenotypic space. But the specialized cells, they might have peaks on some phenotypes because they are specialized, right? I mean, they are more pronounced there. So this is about now the developmental stem cells. For cancers, actually, they put cancers up here. The more malignant the cancers, so they have higher entropy, right? So they are going back to this multitasking behavior, right? So this is a very interesting observation. So what we said here is, let's assume now that the internal state in physiology, it's something that, the internal state entropy in physiology is something that's going down. The second thing is that when we have differentiation, not only the cells are differentiated, but the tissue is maturing. And it means it goes to a unique pattern, right? And these are some, organoid things. And actually what you see here at the beginning, you have a soup of stem cells where actually they're becoming a certain constellation of differentiated cells. And maybe, here you might have an increase of entropy in the meanwhile, because these guys, they have to get differentiated, but then the entropy settles down again lower. So what I want to say is that microenvironmental entropy and internal entropy, both of them, they are evolving during not only physiology and development, but also during a pathology. So this made me think that, and the good thing with entropies is that because they are probabilistic, I mean, information theoretic quantities, we don't need to know all the underlying details. Okay, so they can describe the system in this coarse way. And therefore, we made this, we formulated the cell decision making as a Bayesian learning. So what it means that the cells, actually, they have an internal variable vector, X, and they can read the microenvironment. And the way and then of course, sensing the microenvironment, this can feed back and induce a change in the probability of X. So probability of X is evolving in time. So this is actually the formulation of the Bayesian law, where actually what you see here with red is the empirical likelihood, right? So X is the phenotype, Y is the microenvironment, these are vectors. And then you see here, this is actually the microenvironmental sampling distribution. So the cell has to spend energy to do that, right? And actually with the blue, this is the prior, what we call the prior distribution of X. Or actually, if you think that cells are genetically predisposed to do something, then certainly this prior has that meaning. And if this is just the normalization factor. So in principle, when we have this Bayesian learning synthesis, and actually this is not new, already regarding learning, this perception, these cognitive processes, already Helmholtz, the great physicist, he was talking about this Bayesian perception. But let's overcome this. So the point is that, so I'm saying this 200-year-old concept. The point is that, this is the Bayesian law is static, right? Everything is at the same time. However, my postulate is that the cells try to build optimal priors for the microenvironment because this saves energy. Why is that? Because we know when we have differentiated cells, the differentiated cells know very good about what is happening in the microenvironment. And certainly, they don't have to change their phenotype all the time. Somehow they are in kind of steady state. So what we have assumed is that, after one time step of Bayesian law, the posterior becomes the next time prior. And then if I rewrite a bit the above situation, the above equation, the Bayesian law in this way, you see here, I have something that evolves in time, which is the XT, the probability of XT. And then if I do appropriate upwitchings, I can write an equation about the entropy of this is the entropy rate, or how the entropy increases or decreases of the P of X, right, in different time points. And actually, this is equal to minus the mutual information between internal states and the microenvironment. So in order to evolve this entropy, the system consumes information. And in the very strict Bayesian way, because this is always a positive quantity, this is negative. So actually, when the cell consumes the information, it means that it learns. So it has at the end more pronounced unimodal distributions, and this reduces the entropy of the cell. So this is something that we have formulated some time ago. And in principle, I'm not going to go into the calculations, but what we can do is I can write it in a continuous way, right? And here also, if I continue, if someone wants some more information, I can give him later. We can end up in writing not only the probability distribution, the equilibrium probability distribution, but also a Langevin equation about the phenotypic dynamics. So as you see here, the probability distribution of X at equilibrium, it's like a Gibbs distribution, where this beta, it's kind of inverse temperature, like for the physicists there. But and then here the energy role is being played by the microenvironmental entropy, because this is actually microenvironment conditioned by the current state of the cell, right? And this is actually what we call the least microenvironmental uncertainty principle, because in order to go to equilibrium, as the energy in the physical system is going down, then here the microenvironmental entropy goes down. And we can see it also in dynamic sense, right? I mean, this I can turn it into a Langevin equation, and then I can have my X dot, how it evolves, and then we have this gradient, these microenvironmental gradients that they are driving the system, right? So this is a very interesting formulation that doesn't need the full knowledge of the underlying mechanisms. So now I'm going to show you, I mean, I know I don't have too much time. I have 5, 10 minutes. How many minutes I have, Joshua? Just to be 5 minutes?
Well, you have around 15 minutes.
Okay, good. Then I don't have to rush. I thought it was, I need to rush more. Okay, so good. So then, in order to illustrate that, let us assume that our microenvironmental distribution is a normal distribution, right? So this is the Y, this microenvironment, and X is the internal state. So now the conditional entropy that we saw before, it becomes this nice Gaussian entropy, which is of course this logarithmic case. And then if I plug it into this previous equation, right? So as I said, beta is a parameter. By the way, beta, since now it becomes a parameter which relates to the time scale of this decision making. I forgot to tell you this relates to the time scale of decision making. But but since it becomes a parameter, I can make it positive or negative. And then if you make it positive or negative, the beta, look at here, there is an interesting situation. The steady state distribution, it looks like that, right? So this is like the sensed noise, and this is actually the actual noise. So it is sensed noise into this power, and then this is the actual noise. For beta positive, I get unimodal distributions. So indeed, I go to a reduction of entropy. For beta negative, the steady state, it can be either power law or a multimodal distribution, which actually it increases entropy. So I have a mechanism, this entropic gradient mechanism, it allows us to go to both states. And for the ones that the statisticians out there, if I put beta equal 2, then I will arrive to what is called the Fisher's prior, right? Because this is actually proportional to the 1 over the variance. And this is, a kind of optimal non-informative priors that people will do in Bayesian statistics. So then we can see if the LEUP concept, or the Bayesian learning concept, is helping us to understand tumors, right? This is the big deal. With Yoso, we have worked on this part in bacteria, but we have also worked on something again that we took up again the goal growth part, right? And that I presented you before. And because we knew that this is somehow related to glioblastomas. And then we said, okay, now we assumed again exactly the same model. Like the cell, they have this Markov chain that goes from motile to resting. Resting can proliferate, but then there are the transition probabilities to the corresponding states that they depend on the microenvironment. And then the microenvironment is thus composed from these green and red cells, so motile or resting cells. And then we say this is, so the density of this, so the composition of this microenvironment is actually our Y variable. So then our transition probabilities, they are becoming like that, right? So here I take up this steady state that I was assuming before. Sorry, the steady state distribution that I was assuming before, and I turn it up into transition probability. And then you see these transition probabilities now, they are like sigmoidal functions, right? That they depend on this delta S is actually the change in the microenvironmental distribution when a cell transition is taking place. And then we took this up and we made stochastic simulations using the Langevin equations we had before. And then with this red are the moving cells, the dots are the resting cells and proliferating. And what we have done is, again, we wrote this LEUP now grow model it's a system of reaction diffusion equations, one equation for the resting state, one equation for the motile state. I could turn this in one equation if I put this to be fast, right? This is what I have done before, but I didn't do it. So this switching term now, you see, it encodes the transition probabilities. And interestingly, we found out the bistability that we saw before. So this, let's say, agnostic mechanism was able to reproduce the bistability observed by changing. And this happens, sorry, this is actually the bifurcation diagram. This bistability comes if we play with beta. So for positive beta, increasing that, we had the bistability, where before we had monostability. So somehow we recovered what we knew before. And this was also with Simon that Yoshi knows also very good because they were doing the PhD at the same time. However, can we find any biologically relevant microenvironmental variable? Because again, this is like, okay, very theoretical, the study. So recently, the goal growth concept has been connected to epithelial mesenchymal transition. Actually, they were saying that this is something, let's say, more or less the same story, right? This review, which actually it's very new, it's 2023, and they said that the EMT, the epithelial mesenchymal transition, which actually epithelial cells are cells that they are composing our tissues, right? And usually they are static. I mean, they don't move. Cortical tissues, they are all dissolved. But when there is a wound healing, they go to this mesenchymal state, which is actually invasive cells, motile cells, and then when the wound healing process is over, they go back. So if we assume this is the goal grow now, then there is a mechanism known for this epithelial mesenchymal transition, which is the notch-jagged delta signaling. And in principle, this is not only known that it controls epithelial mesenchymal transition, but very recently, this is 2019, 2017, this Nature Communication paper, I don't remember, they found out that these knots actually in glioblastoma cancers, so you see these are glioblastoma cancers here, this notch is controlling the epithelial state of cells, which is the growth state or the resting state, to migratory state. And also they go to another, if they go lower, the notch goes to something that is a network migratory state, but I don't want to get into that. This is something that's very new. So for me, I'm interested only here. And still the notch is a relevant molecule. So we said, okay, let's do the following. Let's take now the notch delta pathway and compare it with LEUP, compare it with our Bayesian learning. And what we have done, we know that in the market there were very good notch delta jagged models. And this is actually my friend Mohit. He's in India now, but he was in San Diego by that time. He developed this very nice model where actually it controls the notch delta signaling. I mean, a lot of you know that. And the interesting thing with this notch delta jagged signaling is it produces these checkboard patterns. And in principle, this is happening in two ways. So this is called actually lateral inhibition patterns because notches get upregulated and the neighbors downregulate the delta. And this happens when the jack doesn't play a big role in the system. But when the jack is on, it plays a big role, then we have uniform patterns. So we have like a mixture. So, and this is called lateral induction patterns. So the question is, okay, this is actually, these simulations that are being done, if you make a cellular automata and each of the cell, you are solving these four equations. Now, we took our model and we found out, this is actually our agnostic LEUP model, and this is the equations that you saw before. And then we found out that for beta smaller than 0, we had something that it was increasing entropy. So these are the lateral inhibition patterns. And for beta positive, we had something that it was decreasing entropy. So all the cells have the same fate, right? And then here you have mostly bimodal distributions, and here you have unimodal distributions. And interestingly, this is just an observation, but then we made some calculations actually, and then we calculated analytically the beta. So I don't want to go, of course, into that, but this is the result how beta can be calculated from known equations. And then we found out that in this region, where beta is positive, let's say, and this region is beta is negative, then we compared and we saw that the system was bimodal here, and here it was unimodal. And this is actually not the LEUP system, but this is actually the original system, the notch delta system. And to go further than that, we checked also the pattern. And then we found out that, so these are simulations now at all these points, we found out that when we are in this region of beta, that we have positive, I think it's inverse here, but here it should be positive. But anyhow, here when we have in this region, we have this patterning taking place, and in this region we had no patterns. So not only the bimodality was predicted as the steady states that I showed you before, they could predict, but also the patterns, they were compatible with that. And this could be predicted only by the virtue of beta. So beta could really coarse grain even the patterning dynamics, which is very interesting, right? I mean, now I have an agnostic system that I didn't know anything about that, and still I could reproduce the main aspects of the problem. But again, I didn't talk to you about tumors. So in the last part, and now I'm finishing, I have 3 slides more. What I'm going to show you is that if you remember what I told you is that that the cancer cells, they were like stem cells, right? In entropy terms. And in principle, this is not really far away what biology says. In 2012, actually, these two guys, they got a Nobel Prize. So this is the Yamanaka and Gordon. And actually, they were able to turn stem cells into so you could then differentiated cells program them into stem cells, right? So these are what they call the Yamanaka factors. It's very well known. And in principle, this is very interesting because already in this cell paper here, they were saying that the glioblastoma actually, they are emerging from this de-differentiation or this reprogramming process. So these are differentiated glioblastoma cells, and for XY mutation, they go back to a stem cell-like state, and this stem cell-like state in the mature brain is cancer, right? So now we want to say, okay, this is interesting. So how about to see if our theory help us to identify when can we have tumors or not regarding the transition between this stem cell situation and non-stem cell situation. And then when we have done in 2021, it's like we assumed again a theoretical model where we had tumor cells that they had transition probabilities to become stem cells to differentiated cells, right? And then back, this is the inverse micro process. However, these tumor cells, of course, or these cells, they were not living alone, they were living in a community. And then we want to see not what a single cell is doing, but what the community is doing of cells. So this macro state, what I'm calling is here like a community of, I want to see how a community or a tissue, apart from stem cells, they can go to a differentiated tissue and back. So this is the macro process. This is the macro Markov process, right? I have two Markov process, one for the micro world, another for the macro world. And of course, these two are connected by averaging. So here, these are the micro world process, and this is the macro world process. And this P of S now, what is that? This is the probability of a cell being stem cell or pluripotent or differentiated. And here, this is what we got from the equilibrium distributions of LEUP, right? What I showed before. These Gibbs distributions related to the microenvironmental entropy. So this is what I'm doing here. And this is just, here I just assume, this is what I'm going to try to understand. So because these ones actually, I didn't know them exactly, then I assume that there is this Crookes fluctuation theorem, where actually it says that if we have a process that goes from stem cell, that goes one direction, and the other process that goes the other direction, then this happens at the expense of losing some heat. So this is what we're assuming here. We're assuming that when the cells, they are changing from stem cell to differentiated and going back, there is a change in the metabolism. And actually, this is completely true. Indeed, when differentiated cells, when stem cells are eating predominantly glucose, so they have anaerobic metabolism. And in the differentiated state, they have aerobic metabolism where they consume oxygen. And this happens also for the cancer cells, right? So there is a change in metabolism. And actually, this data
You
can
have
be
5
exactly
minutes.
calculated. Yes, I'm finishing. This is like 2 slides, actually. So just to finalize here, what we could do here is we can find probabilities that this W plus is going to a cancer state and W minus is returning from the cancer state. We can here have in this exponent different quantities that they are connected to metabolic changes, cell decisions, to proliferation, of course, into adaptation. And then when this is bigger than 1, then we have something that goes to differentiated tissues. But when this is smaller than 1, then we have reversibility, so cancerization, right? And the interesting thing, all these quantities, I can somehow understand them quantitatively. And they are connected what we call hallmarks of cancer. So this is my last slide. The vision here is that what we want to do is to, again, to go back to this theory and to have this axis here, have three axes, that I can quantify the biological process, and then I can see how can I change this biological process in order to revert this ratio that allows tumor to become back to a normal tissue, right? Where before it happened this one. Of course, this is easier to say than doing that. This is something we try to do, but I would like to stop now. And this is Arnab is one of the guys that he was working a lot on this theory. And he also made this very beautiful picture. And yeah, I would like, thanks for your attention. Hopefully I kept your interest. It was a very long talk, and I'm open to questions.
Thank you for your very nice talk. We have time for some questions. Okay, so maybe I start with a cheeky question. So, do you think it's good to have a growing number of theoretical biologists at the university or not?
If we have, sorry, I didn't listen, it was interrupting.
Oh, sorry. Do you think it's a good thing that there is an increasing number of theoretical biologists?
I mean, I'm a bit biased and I would say yes. But why I would say yes? I would say for two reasons. First of all, theoretical biology is an interdisciplinary field, right? So it's a field that allows different departments coming together. And in principle, it's a kind of study that not only, of course, goes to the application, but goes into interesting math. Because the truth of the matter is that the math we were using until now, we used a lot in physics, and physics has to do with inanimate matter. However, to living systems, the math that we have to use, they're way more challenging, and we need to find solutions. As you see here, I needed to resort to things that, they are not classical. Also, I didn't solve that, but there is another way that all these uncertainties that we have, again, we can deal with them with a combination of dynamic models and machine learning, right? So this is another thing that I'm working, but I couldn't solve it here. So what I want to say is that the mathematical biology or theoretical biology now, it's a field that is very evolved and has different directions. Either can go definitely to application. So this is good for a department, even if you think to bring projects, right? I mean, this is something that makes a department viable. But on the other hand, on the academic side, it offers very new and fresh problems to mathematicians. So I would say, even if I see it from the classical point of view of mathematics, I believe that mathematics at the end would be much more benefited from the interaction with biology and medicine, as it's exactly the same way as physics evolved mathematics. Biology will evolve the field even further.
I also agree that it's good to have theoretical biologists. Any other questions? No. I want to ask you something from your slide. Can you go back a little bit?
Which one? Where?
I think it was the next one.
Ah.
No, the previous one. Yeah. The one with the patterns.
Here? Okay, yeah, here. Cool.
Yeah, here. So here you talk about the modality index, and I'm wondering if this is like a Fourier transform, or is this just a PDF, or what is it?
Okay, yeah, this is the PDF of the phenotypes. This is the PDF of the phenotype. The Fourier transform is here. Actually, this is what you. So what? Yeah. So I went through that, but very fast. So this is the actual pattern. This is the probability distribution of the phenotype, the average phenotype, right? So you see there is a kind of bimodality, but it's a bit more sketchy. But here, what we have done, we took the structure factor, the Fourier transform of the domain. And then what we saw is, maybe I have it here. I think I can go. Yeah, this is it. So look at here. If see, this is actually when we have chessboards. This is the wave number. This is the power spectrum of Fourier transform, and this is how it looks if we have chessboard. If we have random, completely this gray line. So it's something that flattens out. Now, what we have done is we checked if we cross, let's say, this randomness, right? And you see for some simulations, and in this random uh, regime, right? So it means that have uniform patterns. But as you see, for some other simulations, I'm having some peak here, and this peak is following this trend, right? So of course we never have a perfect chessboard. This is the perfect chessboard to have it like that. And therefore, what we have done is we were just comparing a difference from the random to the pattern. And then what we saw here is phase space diagram. It's exactly that. I mean, this is the area and the curve between the random Fourier transform. It is the area and the curve between the actual simulation with Fourier transform. And where this is positive, we have a pattern. Where this is negative, we don't have a pattern. So we have all three
Okay.
information, right? What happens to the phenotype, the microscopic, what happens to the pattern, and of course, this is the real pattern.
All right. It's very interesting to see all this.
And the B, and actually the B, as you see, the B is represented. I mean, it can cause all these behaviors.
Mm. Yeah.
And
Yeah, I think
even
for
now we have
students.
yeah, sorry, yeah, go on.
No, tell me.
No, I just want to say that even now we have some new results that we were trying to understand if we can get reduced models. So if we have fast and slow dynamics, and we identify now in these two regimes, we can get two different reduced models. So here, for instance, nodes dynamics are fast, and here we have delta jagged fast. So it's pretty interesting what's happened biologically even.
Yeah, because this is more static, and now you
Yes,
get more
yes,
dynamic data.
right.
Nice. Yeah, I just want to say that it's nice that the students see that you have here stochastic processes, probability, machine learning, analysis, everything is there, basically.
As I said, it's an interdisciplinary field, and as an interdisciplinary field, definitely it's very challenging mathematically. And, you have to resort in many techniques. But
Mm.
also, this is also another thing that is important. I mean, the mathematical biologist, or theoretical biologist, it's not only someone that works alone. He cannot work alone. He has to work in teams because not, I mean, you cannot have these skills perfect. Not only mathematically, even of course, the biological experiments. So you need to develop a language to communicate with different disciplines, and I think this is actually the most one of the most challenging things, because when we are mathematicians and when we study mathematics, we don't learn that. We learn to do our stuff, and we think sometimes mathematicians are some people that they stay over their desk doing calculations, and you do it, right?
Mm.
But
Yeah.
it's not only that.
Yeah. Well, it's very good for everyone, also for me to hear some things that I didn't know before. So let's give a big German kind of applause to Babis.
Although Greek, right?
Thank you, Babis.
Thanks a lot. Thanks
Thank
a
you
lot
for
for
your
having me
talk.
here, and I hope I got some people inspired to follow the field.
Yes, of course. Thank you, Babis.
Excellent. So have a nice day for you. For me, it's night.
Thank. Yeah, have a good night.